part 2 of chapter 4. Now we're going to look at the cell wall of bacteria. So the cell wall gives the cell its shape. It's semi-rigid. It's not completely rigid, but it's definitely more rigid than the flimsy plasma membrane. And I already showed you how it's the anchorage point for flagella. In addition to the plasma membrane also being an anchorage. Other structures like fimbriae, pili, etc. will also attach at the cell wall. Sometimes certain types of bacteria have capsules, which would be on top of the cell wall. Now, the cell wall is really important for maintaining this separate environment between the inside and outside of the cell, even though ultimately it is the job of the plasma membrane to provide a separate environment. Cell walls of bacteria are always made of peptidoglycan, so we're going to look at that. We have 2 main categories of bacteria. One is called gram-positive and one is called gram-negative. And that's based on the location of the peptidoglycan, which we'll see in a moment. So don't mind this little noise. My throat is very raw from all of these recordings. I accidentally recorded an entire hour that didn't have any sound, so I have been talking for hours now by myself in my office. Okay, so the cell wall keeps the cell from rupturing when the you think about the environment inside the cell is very concentrated solute concentration compared to the solution that it will be suspended in. So water would want to rush in to the cell. So the cell wall helps it resist that osmotic pressure. Also, the first step to identifying a bacterial organism, whether that be for research or for diagnostic, identification of an infection, the first step is always to determine if it's gram-positive or gram-negative. And that's really important. Antibiotics that work for one group typically don't work for the other group, although we do have some what we call broad spectrum antibiotics that work for both. But you always want to give the most specific antibiotic whenever possible to, one, know that you're giving the most effective antibiotic for the infection, but also limiting side effects to the patient. Okay, peptidoglycan is made up of these units called NAG and NAM. NAG and NAM are abbreviations for the full chemical name, which you'll see in your book. I personally don't see why it's important to make you memorize N-acetylglucamate and, whatever. NAG and NAM is good enough in my class. So the monosaccharides, or the small sugars that make up peptidoglycan, are called NAG and NAM. And they're very closely related to glucose, which you could deduce because it's a monosaccharide. Okay, so one unit of them together, right? So 2 monomers put together is a disaccharide. And then we get repeating units of this disaccharide of NAG and NAM to make the actual carbohydrate, which is the backbone of the wall, okay, the cell wall. Each of these carbohydrates are held together by polypeptide chains, and so that's where you get the peptido for peptide and the glycan for sugar. Penicillin actually interferes with the ability to link those polypeptide chains together. And so over time, that bacterial cell wall is going to become very weak and actually give in to osmotic pressure and lyse open. So penicillin or beta-lactam ring antibiotics, that is, they target the cell wall by interfering with the ability to make these peptide chains. So here's the individual units: NAG and NAM, NAG and NAM. And then the blue and green spheres are the peptides that hold the carbohydrates together. And then you layer that. And after lots of layers, you have a very strong wall, which we call peptidoglycan. It's more thick when it's gram-positive, and it'll be thinner when it's gram-negative, and we'll take a look at that right now. So in gram-positive bacteria, they have the thick peptidoglycan cell wall that I was just talking about, but they also have this other substance called teichoic acid. It's actually a type of alcohol and phosphate molecule, and it gives some extra strength to this cell wall, which is rather thick. Okay, it might also play a role in regulating the movement of cations, positive ions, through the cell wall as well. And there's these two types of these teichoic acid: lipoteichoic acid and wall teichoic acid. And we'll see an image of that in a moment. It just has to do with where it's located. Does it span the entire plasma membrane and cell wall, or is it only in the cell wall? That's the difference. I already mentioned this. And okay, so the polysaccharides of the cell wall actually are one of the many antigens. So anything that's on the surface of the cell can be an antigen. Okay, and polysaccharides are the most common of antigens, but we also have proteins. And you're actually probably familiar with this idea because of the pandemic, and you've been learning probably about the vaccine, perhaps, and how we need to target the antigens on the virus so that we can make antibodies. It's the same concept with the bacteria. We, our immune system will recognize a surface molecule called an antigen, and it'll make an antibody in response. It recognizes it as being foreign. If it was not foreign, if it was self, like we have antigens on all of our cells, but we know that it's us, and we don't make antibodies against it unless you have an autoimmune disorder. Okay, so you notice, as I said, the wall teichoic acid does not span into the plasma membrane, whereas the lipoteichoic acid does. So that's the difference. So extra strength and support for the thick cell wall, and another way to say cell wall is peptidoglycan. Okay, so this is gram-positive. You have the plasma membrane, and you have the thick cell wall. All right, now we're ready to look at gram-negative. So gram-negative bacteria have this additional membrane called the outer membrane. And it turns out, because, I mean, I don't know why, but we notice that they have an outer membrane, and then their cell wall is actually thin, and then they have their plasma membrane. It also turns out that they have their own molecule for support and strength called LPS. And LPS is a major antigen responsible for things like an overreaction of the immune system. So you'll read about that in your book. Okay. The space in between each of these layers. So the space between the outer membrane and the plasma membrane is called the periplasm. And we'll see this in the image on the next slide. Having an outer membrane does offer some extra protection, a little bit of protection from the immune system, not as much as a capsule, and a little bit from antibiotics. Like penicillin doesn't really work on gram-negative bacteria. And we also have these special poron proteins that create channels that go all the way through, so that the cell can regulate the movement of substances all the way through from inside to outside of the cell. Okay, so here's an image of that. You see your plasma membrane, and so far everything looks the same, but now you have a thin layer of peptidoglycan. Now in this image it's showing a single layer. It wouldn't be that thin, but they're just trying to demonstrate in the most simple way possible. And then you have this outer membrane, and you notice all these like tail-like extensions, and those are actually the LPS. So I'm going to show you that on the next slide. It's also shown right here in blue. It's showing part of the LPS molecule called lipid A is embedded in the lipid. And then you have the core polysaccharide immediately after that. And then at the end you have the O polysaccharide, which is a major antigen. And in addition, this lipid A can act as a toxin. So I have that here for you. So lipid A is what we call an endotoxin because it's normally inside of the membrane, the outer membrane. So only when the cell dies or when there's a lot of cells undergoing rapid growth, it's really just those two scenarios that we would even be exposed to lipid A. But then that can act as an endotoxin. And the part that we're normally exposed to is the O antigen. And so O antigen is exposed on the very outer surface of the bacterial cell. Thus, it's a good target or antigen for recognition by the host, us, and antibodies that we make against it. The O antigen is this entire long region, okay, shown in red. You have a small core called the outer core because it's immediate, right? This inner core in the lipid A is inside of the membrane, and the outer core is the first part that's outside of the membrane. Okay, so again, LPS stands for lipopolysaccharide because you have a lipid portion and you have the polysaccharide portion. It's a structural protein, we'll call it, but it's really, lipopolysaccharide. It's both lipid and, has a protein region and it has the saccharide region. So maybe calling it a structural protein is not the best way to describe it, but it is a structural molecule. So how about you replace protein with molecule? It protects the membrane, that outer membrane, quite a bit. I already kind of told you a bit about its toxicity. Yeah, I think that's all I want to tell you about this for now. I've already given you so much information. Okay, so the gram-stain is a set of steps so that you can distinguish whether you have the first type that I showed you, gram-positive, single thick cell wall, single plasma membrane, or if you have gram-negative, which has the outer membrane and the thin wall and then the plasma membrane. And it turns out that just by the nature of the structure of those cells, you will get a different outcome upon staining, and decolorizing. So the very first thing you do when you want to perform a gram stain, and most stains, is what we call heat fixing. You take a slide, you take your bacterial culture, you smear it on your slide, and you let it air dry. Once it's air dried, you wave that through a flame just a few times through the Bunsen burner flame, because you don't want to crack your slide, right? And you don't want to melt your slide, but you do want to get the bacteria to be permanently attached to the slide, which is what we mean by fixing. Then, now that those bacteria are fixed or stuck onto the slide, you can add your dye and you don't have to worry about washing the bacteria away. So the first dye is called the primary, and it's crystal violet. It's a very, very dark purple, and it's good at getting in through the cell wall into the cytoplasm. And so then we let that sit for, when we do the gram stain lab, you'll memorize the details, but for now we just time it, then we rinse it with water, and then we add what we call the mordant, which is iodine. And when the iodine goes through into the cytoplasm, the iodine reacts with the crystal violet and forms these large crystals. And those crystals are now too large to leave. And so when you do this next step called the decolorization step, where you get an acid alcohol and you briefly rinse it, these crystals are going to be trapped inside the gram-positive cell. And the reason for that is that the alcohol causes the peptidoglycan in the gram-positive cells to tighten up, and it actually makes the cell hold on to those crystals even tighter. But it turns out that when you expose gram-negative cells to this alcohol rinse, it rips away their outer membrane, and that leaves holes in their wall, and those crystals can then wash right out. So gram-positive do not become decolorized, and gram-negative do. Now you might be wondering, how does the gram-negative get red? And it looks like it got deleted from my slide, so you can go ahead and add this on. After the decolorization step, we have a secondary stain, which we call a counter stain, and that's with safranin, or a very red stain. And if you add red to purple, you still see purple, so gram-positive will still look purple. But for gram-negative, where all the purple got washed away, when you do your counter stain, all you see is your counter stain. And we have all different, morphologies and arrangements of gram-positive and gram-negative. These just happen to be the examples we're using at this moment. Do not think that whenever you see rods that they're going to be, gram-positive. Not at all. In fact, E. coli, which I think is what we have here, is gram-negative, and it's a rod. This is probably Bacillus subtilis because it's so large. Okay, so we do have these main categories of gram-positive and gram-negative, but we also have some that don't quite fit those rules, like what we call acid-fast cells. These are cell walls that have mycolic acid in addition to the peptidoglycan, and this forms a waxy lipid coat. And the most famous example, and where we get the name, is Mycobacterium, and you know this because of Mycobacterium tuberculosis, which causes the respiratory disease tuberculosis. And there's also another bacteria called Nocardia. This is not so common, but it is, well, it's not such a common infection, but it is a common bacteria. It's in the soil. It can easily be in water as well, and it can infect lungs. It can even infect the brain and the skin. So people with a weak immune system, such as someone who's undergoing some type of like chemotherapy or organ transplant, or any reason that you have to suppress someone's immune system, or in the case of someone who has AIDS, advanced form of having a HIV virus. All of these, any reason that someone has a weakened immune system. Elderly have weakened immune systems, and if they have diabetes or any other chronic illness, that makes them even more vulnerable. So anyways, this does cause infections, but more than likely you and I would not suffer from this. Okay, so anyways, if you were to remove this mycolic acid layer, then the cell would behave just like a gram-positive when you do a gram stain. But if the mycolic acid layer is intact, then you would end up with a gram stain that looks both positive and negative, and you would be confused. Another reason why it's so important to always have your positive and negative control is so that if you do encounter an acid-fast organism, you'll know, instead of questioning your technique of your gram stain. When you do a gram stain, and I can't recall if they show this in the gram stain labster, but I think they do. You would have your positive control, your test organism, and your negative control. It doesn't really matter which one's positive and which one's negative, as long as you know. And so a lot of times you can label a little plus and a little minus with like a wax crayon, a wax pen. And you might even draw a circle so that you know where you put your specimen. And if your positive looks like a true positive and your negative looks like a true negative, meaning that this is purple and this is red, then if you have a mixture of purple and red in your test, you would say, I probably have mycolic acid going on. And then you would know to do another stain. Whoops, that came out crooked. This is supposed to be a slide. You would do another stain called the acid-fast stain. It has a different set of reagents. The primary stain is called carbofusion. It's good at binding to cytoplasm. And then when you do your acid alcohol rinse, it's not going to decolorize it, and that's why we call it acid fast. So these would stay the red color, that the carbofusion is red, much like the color that I'm using right now, actually. And so if your test, comes out red, then you have an acid fast organism, probably Mycobacterium, as long as your controls, of course, are correct. And so your negative for acid fast is actually blue, which is the counter stain. Okay, so that's the acid fast stain. You can see examples in the steps of the acid fast test in chapter 3. All right, next we have mycoplasmas. There are some of the smallest cells known to Earth, and they lack cell walls, and that's what we call mycoplasmas. Not a whole lot of bacteria in this group. We have about 15 species that cause human disease. We have other species that are saprotrophic, meaning they're decomposers. And that's about it. So it's all one genus called Mycoplasma, and then, 30 or 50 species. And that's it. So not super common. But it turns out, 15 of those species can cause human disease. Okay. So still important to know. Okay, great. Okay. And then furthermore, I want to remind you that archaea do not have cell walls made of peptidoglycan. Only bacteria have cell walls made of peptidoglycan. So archaea can have cell walls made of pseudomurin, something kind of similar, but not the same. And then other archaea don't have cell walls at all. Okay. Lysozyme is an enzyme we make in our tears and certain other areas of the body, and it's able to digest the disaccharide NAG/NAM and break down the cell wall. So this is why we don't get eye. This is one of the reasons we don't get eye infections all the time. There's microbes in the air, and our eyeballs are exposed to the air constantly, and our tears are constantly wiping away bacteria and viruses that land on our eyes. And we have the enzyme that helps kill it, along with just the mechanical rinsing away. Okay, and I already told you about penicillin. So please make sure that you're understanding and remembering the material that we've covered so far. Like, why are capsules so important? How do bacteria move? Would you be able to identify them under the microscope? And then also thinking about cell walls and thinking about antibiotics or drugs that target cell wall synthesis. So how does penicillin, for example, kill gram-positive bacteria? Why doesn't that work on gram-negative? I think it's very important when you study that you start getting in the habit of making like these summaries, and compare and contrast summaries are really helpful. So I've gotten you started here. I talked about penicillin, and I did not talk about tetracycline. We're not going to go into the mechanism of tetracycline right now, but just remember that this is an antibiotic that works to kill gram-negative bacteria, whereas penicillin will work to kill gram-positive. And you do know a little bit about the mechanism for penicillin. We already talked about that. We also talked about why gram-positive cells retain crystal violet and why gram-negatives don't. Okay? So now we're going to talk about structures internal to the cell wall, still talking about prokaryotes. So inside of the cell wall is the plasma membrane. Every single cell on Earth has a plasma membrane. Cells may or may not have a cell wall, but they always have a cell membrane. Okay? The cell membrane is called the plasma membrane. These are made up of lipids. So the most simple form of looking at these phospholipids is if you were to just take a handful of phospholipids and throw it in some water, you would get all these what we call micelles that would form, where the heads, which are the phosphate heads, would face or orient towards the aqueous solution, and all the fatty acid hydrophobic tails would huddle inside to stay away from the aqueous solution. This is natural. It's energetically favorable due to the chemistry of the heads and tails. This property also helps us understand how membranes form and behave, and it also leads us to this fluid mosaic model. This is a very important video. It's also short, less than 2 minutes. Watch it. It will explain the fluid mosaic model. So our plasma membranes are 2 layers of phospholipids where the tails face each other and the heads face the outside. So this space up here would be like the outside of the cell, whereas this space down here would be the inside of the cell. Just like you see in this image, inside of the cell, outside of the cell. But notice if you only have the phospholipids, they start to kind of get a little out of hand. Some of them are close together, some of them are far apart. They're not having a very super organized structure. So by adding some complex lipids called hopanoids in between, you can actually get nicer, more organized structure, kind of tidy it up a bit. But there's still fluid in that you can, things like these proteins can actually move around in this membrane. Okay, so the membrane is not rigid, but it is a little more organized by having these extra small lipids inserted throughout, which are shown here in red. So just periodically. You also notice there's all sorts of stuff sticking through the membrane, right? The cell membrane does a lot to regulate the function of the cell and to regulate what goes in and what goes out. So we have signals that can land on the outside where the signal cannot go through the membrane, but by landing and attaching to some surface protein will cause some action to happen inside the cell. Other times we have molecules that actually go through protein channels and go all the way into the cell. So all sorts of stuff going on in the membrane. So we have the selective permeability, selecting what goes in and what goes out and what does not get to pass through. We already talked about this, but you can damage a membrane by exposing it to alcohol. Okay. Other types of detergents can also do this, and certain antibiotics can as well. So the reason hand sanitizer works is because it's made up of 70% or up to 95% isopropyl alcohol, which is rubbing alcohol. You could use a different type of alcohol, but this is cheap and readily available. When we work in the lab, we use ethanol. It's not pure, it's lab grade. It has toxic substances that haven't been removed, but don't need to be removed because of the application. So if you were going to, you're thinking ethanol, it's what you use in cocktails or whatever, that would be purified ethanol. So never ever drink lab ethanol. Rubbing alcohol too, you wouldn't drink it, just the smell alone would stop you. But if you want it to be antibacterial, if you want it to disrupt membranes of bacteria, it needs to be at least 70% strong and no stronger than 95%. You need a little bit of alcohol, otherwise it evaporates so fast that it wouldn't have time to do any harm. Okay, so movement through the membrane. You've probably already learned about these processes in your intro biology class, but we will review them. So simple diffusion means that a molecule can move directly through the membrane, and it will always do this from the concentrated solute to less concentrated. So movement of a solute from an area of high concentration to an area of low concentration directly through the membrane. And because of this concentration gradient, you do not need any ATP. So this is called simple diffusion. Osmosis can be simple or it can be facilitated. So some water can go directly through the membrane, Whereas you also have these special channels called aquaporons that allow for faster, more water movement. And so osmosis refers to specifically water moving through the membrane. It doesn't tell you whether it's happening directly or through a channel. Okay. Osmotic pressure would be the amount of pressure you would need to exert to stop water from moving through the membrane. And remember that water is always going to want to move towards the more concentrated solute to create an equilibrium. And usually the solute is trapped and not able to go through the membrane itself. And so here is a simple experiment that we do in pretty much every biolab. And so you may well have done this in high school, and even if you took a Bio 21 here. We have a dialysis tubing, which here I think I'm just referring to as a sac. And they refer to it as a cellophane. Okay. Same thing. It's a membrane that only allows water to get through. Okay. You can have a fancy stopper with a glass tube that goes through it, or what we tend to do in labs, simpler. We just tie a knot, close it off, and we weigh it before and after we put it in the beaker. And what you'll notice is that inside of the tubing or the cellophane, you have a bunch of sugar shown in purple. And then you're just immersing this bag in a glass of water. And then you leave that for a couple hours. You'll see the bag got bigger because water flowed in. The solute, being the sugar, is too big and it is not able to get out. So the only way for equilibrium to be reached is for water to flow in. So the solute's too big to move out, so water will flow in. And remember, the cell prevents this from happening, or prevents it from bursting open by having a cell wall. Good. So here is the before and here's the after. You can also see not only has the bag gotten bigger, but the level here has also risen. This level would just provide a measurement instead of having to take the bag out and weigh it on a scale like we do in our labs. This lab is usually done in the very beginning so that you have the 2 to 3 hours to let this happen while you do other things. You probably remember this. Okay. So if molecules are going through a protein channel, we call it facilitated. As long as it's still flowing down the concentration gradient from high concentration to low, then it's still no energy required. Okay, it's still diffusion. So facilitated diffusion, solute will combine with a transporter protein in the membrane to travel from one side to the other because it's either too big or too charged to go directly through. So notice in this image, we have these positive ions or this positive molecule that's not able to go through the membrane. It has to go through a channel. Here we have a large molecule, glucose, that has to go through a channel. And here it binds and then it gets through. And here it looks like it just slides through. So this is also demonstrating that we have different types of protein transporters. Some that act like a channel and some that act like a machine or a lever. In either case, it is going from high concentration to low concentration, which is why it's called diffusion. So no ATP, but it is using the help of a protein. Okay. Then the third category is active transport. It requires ATP because it's active. It also requires a transporter protein, and it's going against the concentration gradient. So we want to get all of our glucose. So at some point, we can do active transport for glucose, for the remaining glucose. There's also other substances that we want to be greedy with, like you'll learn about the sodium-potassium channel, and we'll do work to maintain a very, very steep gradient of potassium inside the cell. Okay, so here it doesn't matter what we call these substances, but just that we have a lot of them inside already, and we're trying to transport the rest of them inside. So we have to use ATP to forcibly shove them inside against the concentration gradient. And so in summary, you have diffusion straight through the membrane and down the concentration gradient. You have facilitated diffusion through a protein, but still down the concentration gradient. And so these are passive transport because they're types of diffusion going down the concentration gradient. That's in contrast to active transport, which requires ATP so that you can go from low to high, go against the concentration gradient. So how are simple diffusion and facilitated diffusion similar? How are they different? And lastly, compare and contrast facilitated diffusion with active transport. Inside of the plasma membrane, you'll find a semi-fluid substance called the cytoplasm, and everything inside the cell is suspended in the cytoplasm. We don't have a nucleus because we don't have membrane-bound organelles, but we do have a region where you'll find the DNA, and we call that the nucleoid. This is where the bacterial chromosome hangs out. Usually at least one end or one part of the chromosome might be anchored to the plasma membrane to keep it in that place. Regardless, the bacterial chromosome is circular. It's still double-stranded DNA. It's the genetic material of the cell, but it is circular. So double-stranded continuous strand. An image you can see right here. Now they're not showing it bound to the membrane, and that's fine. But you do see that it's in a dense, the DNA chromosome is densely kind of wrapped up a bit. If you were to unravel that, it'd be one big circle. Okay. They're showing all the things we talked about, or the majority of the things that we talked about, but you wouldn't necessarily find all of these structures on every organism, right? There's one thing we didn't talk about yet, and that's the plasmid. The plasmid is actually what is shared through the pilus when we talked about conjugation. I'm sure you're familiar with these parts that we haven't talked about yet, but I will briefly review them now. So ribosomes, which are shown here as just all these dots. Cells are packed full of ribosomes because this is how you make protein, right? So the ribosome is used in protein synthesis, and the bacterial ribosome is smaller than the eukaryotic, and so we call it the 70S ribosome. It's made up of a small subunit and a large subunit. The small subunit is called the 30S subunit, and the large subunit is called the 50S. Now, you do not add these units together. Okay, you do not put 30 plus 50 because it totals 70. I know it's weird, but it's because these are units of centrifugation. They're not units of weight, okay? So not additive. But these two pieces come together, and what goes in between them in this space you see here would be the transcript to go from the transcript mRNA, and then this ribosome uses that template to insert amino acids to make protein. Okay. Antibiotics can target bacterial ribosomes. Some of them will target the small unit, some will target the large. Streptomycin, very common antibiotic, targets 30S. Erythromycin, also very common, targets 50S. If you shut down the bacteria's ability to make proteins, it will slowly die. Inclusions are vesicles inside the cell that can store materials, so emergency reserve deposits. There's some that store phosphate, some that store sugars, others lipids, and others might be a gas, such as air. Just if you have a aquatic organism, it needs to be able to get close to the surface and also farther away from the surface of the water. And by increasing or decreasing gas, it can change its buoyancy. Okay, so that is, one more topic about bacteria is endospores. And then we'll be done with the prokaryotes. Okay, endospores are the most resistant structure. They will not, you can boil them and they will not die. Okay, the only way to kill these, there's a few types of chemicals that can kill them. And then really it's just using the autoclave. High pressure and moist heat. And that's why we have to hold that autoclave cycle at that high heat, high pressure for, that 15 minutes at least, minimum 15 minutes, so that we can ensure that if there were any endospores present, that we have actually penetrated through and killed the organism. Okay, Bacillus forms endospores. We saw Bacillus anthracis in your first lab. Clostridium also forms endospores. Clostridium we'll talk about when we do our overview of diseases, but there's several types of Clostridium that cause human disease, and they're all significant. Vegetative growth is normal cell division of the bacteria, but if the bacteria becomes exposed to harsh conditions, it's running out of food or any other harsh condition, it could trigger it to sporulate. Sporulation is the process of making the endospore. The endospore itself is it's a resistant structure for survival. It makes it dormant. It no longer is vegetative if it has an endospore. When the environment is good again, it can germinate and become a vegetative cell once again. This is another short video that goes over the steps of sporulation and germination. Make sure you watch it. This is an image of an endospore. So you see the bacterial cell, not really even the full cell anymore, but mostly just the DNA, a little bit of cytoplasm. The majority of the cell has been ridden of for survival. And then you just see layers after layers after layers of the endospore. And that will again be shown in that video. But this is a real image of Bacillus subtilis. And you can see how thick and just why endospores are so incredibly resistant. So hopefully you found all of this really interesting and exciting. Please tell me, where's the DNA located in a bacterial cell? What's the general function of inclusions? Under what conditions would endospores form? How do you kill them? All right. Good job, and I'll see you for part 3 tomorrow.